Today's session is all about cells. So the first thing that we're going to talk about with cells today is an idea that tends to show up on just about every AP exam in some form or other. And it's this idea of the endosymbiotic hypothesis. So you guys know that really all the cells on Earth today can be divided up into two types: prokaryote or prokaryotic and eukaryotic. And hopefully that you know that prokaryotic cells are bacteria-like organisms. They don't have a nucleus that's enclosed by a membrane. They also don't have the membrane-bound organelles like mitochondria and chloroplast and things like that. Eukaryotic cells are all the other kinds of cells on Earth today. That includes plants, that includes animals, protists, fungi. They all have an enclosed nucleus where their DNA is at least most of it, and also membrane-bound organelles. So scientists think that the first living things on Earth were prokaryotic cells, bacteria-like cells. So the endosymbiotic hypothesis is essentially a hypothesis to explain how eukaryotic cells somehow evolved from prokaryotic ancestors. So this idea was formulated in the 1960s by a lady named Lynn Margulis. And at first, it sort of got laughed at by most of the scientific community. They thought it was a little bit ridiculous. But then they started looking at the evidence and they changed their tune pretty quickly. But the idea is that some prokaryotes began to live inside other prokaryotes in symbiotic relationships, relationships that were good for both, usually mutualistic relationships. Hopefully you know from ecology that mutualism is a symbiotic relationship where both of the partners benefit. So over time, apparently these mutualistic relationships were so beneficial that the two prokaryotes that were living together eventually couldn't live apart anymore. The survival advantage was just so big. And that the ones living inside the bigger ones ultimately became organelles, membrane-bound organelles, because they had a membrane to begin with. So what the smaller ones benefited with is they got protection from the environment, and the bigger ones gained things like energy production or maybe motility, movement. And then over time, due to things like genetic annealing, where some of the DNA segments were swapped between the prokaryotes, this became a permanent relationship. There's several pieces of evidence for this theory. And a lot of the times when we're talking about evidence, we talk about mitochondria and chloroplast. First of all, mitochondria and chloroplast have their own DNA, separate from that in the nucleus. And that DNA is a single circular loop of DNA, single circular chromosome. It's shaped and sized in a way very similar to what we would find in bacteria. So one single circular chromosome. Also, both mitochondria and chloroplast have their own ribosomes, separate from those found in the rest of the cell. And the ribosomes in the mitochondria and the chloroplast are very similar in structure to those found in bacteria, and not so similar to the ones found in the rest of the cell. Something else kind of cool, mitochondria and chloroplast can independently reproduce, even when the main cell isn't reproducing. And they do this in a method that looks almost just like binary fission, or what we sometimes call prokaryotic fission. It's the way bacteria reproduce. And then one other thing, mitochondria and chloroplast, instead of having just one double layer of phospholipids on the outside, they have two. They have what we call a double phospholipid bilayer. So scientists think that this is maybe evidence that the original prokaryote had its phospholipid bilayer, and then once it got phagocytized, pulled in, it got another layer put around it, almost like a vacuole. So there's lots of evidence to show that maybe this could have happened. But the big idea is that in the endosymbiotic hypothesis seeks to explain how eukaryotic cells evolved from prokaryotic ancestors. And the idea is that some prokaryotes began living inside others. They both benefited. And that over time, this beneficial mutualistic relationship became so important that they couldn't separate anymore. And that the smaller ones living inside ultimately became organelles, membrane-bound organelles, like mitochondria and chloroplast, and we could go on with the list of organelles. Any questions there on the endosymbiotic hypothesis? All right, let's stay put here on this screen. This is just kind of showing you how it might have happened. So we've got a big prokaryote. And then here we have some kind of aerobic bacteria, which was a separate living thing. It got taken into this bigger cell. Ultimately, over time, that bacteria evolved into becoming the mitochondria. Here we see a photosynthetic bacterium that got taken into this bigger cell. Over time, those bacteria evolved into chloroplast in plants or algae, that kind of thing. All right, so let's go a little bit further here. Bear with me for a second. My computer's locking up on me here, guys. All right, so another thing that we want to talk about completely separate from the whole endosymbiotic hypothesis is the idea of surface area to volume ratio. I want you to think of the surface area of a cell like the membrane of the cell. And remember, the membrane's main job is to control what can come in and what can go out of the cell. And I want you to think of the volume of the cell like the internal contents, everything inside. Cytoplasm, organelles, everything inside the cell. Here's a really key statement. As cells grow, the surface area to volume ratio for the cell decreases. So as cells get bigger, the volume, at least proportionally speaking, grows faster than the surface area does. That means the insides grow proportionally faster than the cell's ability to move things in and out. So what that means, another important statement right here, as the cells get larger, it gets harder and harder for them to transport in and out the things they need to transport in and out. This is the main reason that cells are very, very tiny. They don't get very big because when they do, they can't transport enough stuff in or out to keep them alive. So smaller cells have a high surface area compared to volume ratio, and they're better able to transport things. Large cells have a lower surface area to volume ratio, and they can't transport in enough nutrients, can't transport in enough oxygen, they can't transport out enough waste to survive. So cells need a large surface area to volume ratio to be efficient. And the main way to do that is just to stay small. So here we see a section off of your AP Biology formula sheet. So this will be on the formula sheet that you can use throughout the whole exam. They wouldn't put it there if they didn't want you to use it. So they're probably going to ask you to calculate some surface areas and volumes. If you can do basic math, you can do this. Not hard. So they're going to ask you to calculate a surface area, a volume, and a surface area to volume ratio. So let's go to the next slide, and then we'll come back and look at the formulas in a minute. So I want you guys now to switch over to your Pear Deck screen and go to this slide, which I will navigate to myself. So let me get you on this slide in Pear Deck.
Yeah, we can't actually move the slides forward ourselves. We're completely.
Yeah, I'm moving you there.
Okay.
I'm getting you there. So here's the slide that I want you on. So everybody should be able to see this now, surface area to volume ratio problems. And notice that it's asking us about a cube, and it's also asking us about a sphere. So if we go back a slide, notice that we have a formula here for the volume of a sphere. We have a formula for the surface area of a sphere. We have a formula for the surface area of a cube. And on this older version, for some reason, there wasn't a formula for the volume of a cube. That's now been corrected. But I'm going to write these formulas for you. Actually, I'll just tell you what the formulas are here. So for a cube, I want you to go ahead and calculate for me. Let's just do the cube first. So I want you guys to calculate for me the surface area and the volume of the cube. And then if you know how to calculate the surface area to volume ratio for that cubic cell, do that as well. And you can do that by writing on your screen. Remember that to find the surface area of a cube, you take the side times the side, and you multiply that by 6. So it's side squared times 6. That's for a cube, for the cube's surface area. And who can tell me how you find the volume of a cube? Just unmute and tell me. How do you find the volume of a cube? Somebody out there I know knows how to find the volume of a cube. Somebody chime in and tell me.
You cube the side length?
Yeah, so side cubed, or you can think of it as length times width times height. Either way works. So side cubed or length times width times height. It's going to give you the same thing.
The surface
So go
area?
ahead on your screen and draw in the calculation for the surface area of that cube.
How do you find the surface area?
For the volume of that cube, side squared. So just side times side. Also, go ahead and find the surface area to volume ratio if you know how to do that. And you can write those calculations out anywhere on your screen. I'll give you about a minute, minute and a half to do that, and then I will go back to the PowerPoint and work through it for you. All right, I'm just going to pull up what you guys are thinking at this point. Oops. All right, so I see several calculations there. Not many surface area to volume calculations. So let's go back to the Zoom screen now. Probably the main thing you need to be able to do is get a surface area to volume ratio. So I'm going to show you how to do all that here on this slide. So first of all, up here at the top, I'm going to calculate my surface area of my cube. And remember, that's side times side, or side squared, times 6. So that would be 0.4. And this is the symbol for a micrometer, times 0.4 times 6, because there's 6 sides in a cube. And when you do that, do the math on that, you end up with a surface area of 0.96, 0.96. And the units would be micrometers times micrometers, or micrometers squared. For the volume, remember it's side cubed or length times width times height. I'm just going to go side cubed. So that would be 0.4 micrometers cubed. And when you do the math on that, that comes up to be 0.064. And that would be micrometers to the third power, micrometers cubed. So in this case, your surface area to volume ratio, and we use a colon to mean 2, right? So surface area to volume ratio would be surface area of 0.96 to a volume of 0.064. Now that's really hard to interpret. So what I would recommend you doing to get a more understandable surface area to volume ratio is to divide both of these numbers by the smaller number. So divide 0.064 by 0.064. Divide 0.96 by 0.064. So divide both this and this by the smaller number. And when you do that, you end up with a surface area to volume ratio of 15 to 1. Now by itself, that doesn't tell you much. This is for the cube. So we found a surface area, we found a volume, and we found a surface area to volume ratio. Let's do the same thing now for the sphere. So to find the surface area of a sphere, the formula is 4 pi times r squared. So in this case, 4 times pi, and our radius in our problem is 0.6 micrometers. So times 0.6 micrometers. We got to square that. So do the math. So it'd be 4, and you can just round pi to 3.14 in this case. So 4 times 3.14 times 0.6 squared, and you end up with a surface area of 4.52. And that's micrometers squared. All right, you can do the same thing now with the volume. For the volume for a sphere, it's 4/3 pi r cubed. So in this case, it would be 4/3 times pi times 0.6 micrometers cubed. All right, so cube 0.6 multiplied by pi, 3.14, multiplied by 4/3. And when you do that, you end up with a volume for the sphere of 0.904 micrometers cubed. So your surface area to volume ratio for the sphere rather is the surface area 4.52 to the volume 0.904. And then to get that in a more understandable number, divide both sides by the smaller number. So 0.904 divided by 0.904 and 4.52 divided by 0.904. And when you do that, you get a surface area to volume ratio of 5 to 1. So for the sphere, this side, 5 to 1. For the cube, 15 to 1. And that leads us to the last question up here. Which cell is more efficient at transporting materials into or in and out of the cell? You guys tell me, which one of these would be more efficient at transporting things? Cube or sphere?
The cube.
The cube. So remember, higher surface area to volume ratio, more efficient at transporting things. And in this case, the cube is more efficient, not because it's a cube, but because it's smaller than a sphere. That's really what makes it more efficient. Smaller things typically have a higher surface area to volume ratio. So there are some other ways to go about getting a high surface area to volume ratio. And some of those are the ways the cell is shaped. For example, there's a lot of structures and a lot of cells that are all folded on the outside. Maybe something like that. And when you have all these folds, you increase the surface area without increasing the volume very much. So we see that in the mitochondria, inside the inner mitochondrial membrane. We see it in the chloroplast. You also will see the same kind of folding in your the lining of your stomach, and also the lining of your small intestine, where you get these little structures called villi and microvilli. So folds, increase the surface area without increasing the volume. Another way to get more surface area is to have, instead of having a cell that's a cube, have a cell that's very long, skinny. Those cells tend to have a higher surface area to volume ratio than the shorter, thicker ones do. So stay small, have folds, or be sort of long and skinny, all three ways to increase the surface area to volume ratio. Now, in addition to this ratio being really important for transporting things in and out, it's also an important thing to look at when you're talking about heat exchange with the environment. Let's take endothermic organisms for a minute. Who can tell me what an endotherm is? Can anyone unmute and tell me what an endotherm is? Nobody? So endotherms are what most of us would call warm-blooded organisms. So these are things that regulate their internal body temperature, like you, like mammals, like birds. And the way we do that is that as we lose heat, we generate more body heat by cell respiration, essentially, to replace that lost heat. So we keep a constant internal environment. Small organism, let's take like a tiny, tiny mouse. They have a really high surface area to volume ratio, that mouse does. We're thinking about their whole body, which means they're losing a lot of heat to the environment. And therefore, to maintain a constant body temperature, their metabolic rate per cell has to be crazy high. Take a blue whale on the other extreme. Very, very huge. Its body has a very low surface area to volume ratio. It doesn't lose heat very fast per cell. So its rate of cell respiration, its rate of metabolism can be slower. What I'm saying is small mammals, small birds, have to have really high rates of respiration, really high rates of metabolism, because they're losing heat really fast. And to maintain their constant internal body temperature, that means they have to generate a lot of heat really fast. Bigger organisms, slower metabolic rate, slower rate of cell respiration, because per cell they're losing heat at a lot slower rate, and therefore they don't have to generate heat at a fast rate. So two things to think about: moving things into and out of a cell, and then transporting heat from an organism out into the environment, or vice versa. Surface area to volume ratio, critically important. Make sure you can calculate surface areas, you can calculate volumes, you can calculate a surface area to volume ratio, and you can interpret those. All right, so we'll stay here on the PowerPoint screen for a little while. We're also going to talk about transporting things into and out of a cell. You guys, I hope, know the cell membrane is made of what's called the phospholipid bilayer, and that things that are ionic or polar, things that are charged, and things that are large, they can't move through that phospholipid bilayer very well. There has to be another way in for them. They get in with the help of either what are called channel or carrier proteins, proteins that extend through that oily phospholipid bilayer and essentially act almost like tunnels to move those things through. Now, things that are small or things that are not charged, not polar, those can tend to enter and exit wherever they want to. They can go in and out straight through the phospholipid bilayer. So we'll deal with both of those things as we talk about different ways cells transport materials. One type of transport in cells called passive transport. This kind of transport doesn't require the cell to use its own energy. It's like its own ATP. It does take energy for this to happen, but that energy comes from the environment, usually heat in the environment. The types of passive transport that we deal with in AP Bio are diffusion, osmosis, and facilitated diffusion. So diffusion is probably the most simple case. This is when materials simply flow from where there's a higher concentration of them to where there's a lower concentration of them. They're always moving from high to low concentration. Eventually, if nothing from the external environment happens, you're going to reach equilibrium. The concentration on each side of that membrane is going to balance out. When they do, that doesn't mean the particles stop moving, but it means that net diffusion ceases, which essentially means if two molecules go from outside to inside the cell, then on average, at the same time, two go from inside to outside. So once they get to equilibrium, they pretty much stay there because it's random. It's about random movement. Diffusion is the way the majority of materials in your body are transported. And again, the biggest benefit of diffusion is that it doesn't require metabolic energy. The cell doesn't have to use its own ATP to make this happen. It's powered by heat from the environment. So there we see our membrane. Again, it's 2 layers of phospholipids. Here's what each phospholipid looks like. And the things that can cross easily are things that are small and or non-polar, not charged. The things that can't get through very easily are large molecules, ions, or molecules that are pretty polar. Those can't get through very well, and they have to have a protein to help transport them across. All right, so let's talk osmosis, because it's the one that we talk a lot about in AP Bio, and it's the one that confuses people. Osmosis is the diffusion of water. So osmosis rather, is a type of diffusion, which means that in terms of the water moves from high water concentration to low water concentration. But oftentimes we define it not in terms of the water, but in terms of the stuff dissolved in the water. Things dissolved in water are called solutes. So we might say that water moves from areas of low solute concentration to areas of high solute concentration. Now realize that if there's a low concentration of solute, that's just another way of saying there's a high concentration of water. And if we say high solute concentration, that's just another way of saying low water concentration. So we're not changing the definition. We're just using different words here to mean the same thing. The definition you're usually going to see used in AP Bio is the movement of water from areas of low solute concentration to areas of high solute concentration. There's yet another way to define it. We're going to talk about 4 different ways really to define it. Sometimes we use these words, hypo and hypertonic. Somebody tell me what hypo means, H-Y-P-O.
Low.
Low, less than. And hyper means high or more than. So hypotonic is just another way of saying a low solute concentration. And hypertonic is just another way of saying a high solute concentration. So we can define osmosis is the flow of water from hypotonic to hypertonic. So 3 different ways of saying the same thing. Don't let that confuse you. Another thing I'll just mention briefly, even though water is small and it probably ought to be able to cross the membrane on its own, there are some special proteins called aquaporins which help water to move across the membrane in facilitated diffusion. So if you see that word aquaporin, just think aqua means water. They're proteins that help to transport water across the membrane, only water. Also remember that osmosis is really crucial for all cells. If a cell doesn't have enough water, it's going to die. If it has too much water, it's going to die. So we often refer to what's called osmoregulation, which is the process of regulating both a solute and water concentration in a cell or in the body overall. The urinary system, that's its main job, is osmoregulation, controlling the concentration of solutes and water in the cells and in the body. All right, I'm going to skip through those next couple of words here. And I want to talk next about what's called water potential. Because this is a math calculation that you might see on the exam, and I want to make sure you can do it. So here you see a formula that's on your AP biology sheet, your formula sheet. You'll have this formula, so you don't have to memorize it. This symbol sort of looks like Poseidon's trident is called psi. It's a Greek letter psi, and it stands for water potential. Think of water potential as water's potential to move. Water always moves from areas of high water potential to areas of low water potential. So that's a 4th way of defining osmosis. Let's backtrack and talk about all 4 again. Most basic definition of osmosis, movement of water from areas of high concentration of water to areas of low concentration of water. Definition 2, movement of water from areas of low solute concentration to areas of high solute concentration. Definition 3, movement of water from hypotonic areas to hypertonic areas. And then finally, definition 4, movement of water from areas of high water potential to areas of low water potential. Now notice this water potential formula has 2 components. One of them is called the solute potential. And that's really what we've talked about so far in our other 3 definitions of osmosis, the solutes. What we haven't so much talked about is this other one, and this is called pressure potential. This becomes really important, especially in cells that have walls. It turns out most cells have walls. Bacterial cells have walls. Plant cells have walls. Fungal cells have walls, and even some protist cells have walls. So most cells have walls, so the pressure can be important. So the overall water potential, which sometimes you'll see notated as psi sub T, is equal to the pressure potential plus the solute potential. And I'm going to talk to you about how to get both of those numbers here on the next slide. So first of all, if we only accounted for solutes, we would deduce that water would move into a plant cell forever and it would never stop. Because inside plant cells are sugars and salts. And if we're talking about a freshwater plant, out here in the container, there's pretty much no solutes. So we'll say 0 solutes in the container. There are solutes here. And if we're just using our solutes, water's going to move from where there's less solutes to where there's more solutes. So according to that definition, water would never stop going into this cell. We know that doesn't happen. And it doesn't happen because as more water goes in, the pressure inside that cell goes up. And eventually there becomes so much pressure inside the cell pushing out that even though water tries to go in because of the solutes, it can't. So the pressure overrides, the pressure potential essentially cancels out the solute potential. So when you're thinking about water potential, you've got to take into account both solutes, dissolved substances, and pressure. And let's look at how those calculations for these things work. So here's some important things to know about water potential. First of all, the water potential of pure water just in an open container is 0. So the water potential of just pure water, 0. And this is because there's no solutes, so the pressure potential 0. And the only pressure acting on that open container is atmospheric pressure, just regular old air pressure. We cancel that out. In most of the problems you're going to see in an AP biology class, the pressure potential starts out as 0 in most cases. Unless they tell you otherwise, make the assumption that the pressure potential is 0. And then as more and more water goes into that cell, that pressure goes up. It builds as more water enters the cell. There's a formula given to you on your formula sheet to calculate the solute potential. And I'm going to write it a little bit bigger here so you can see it. So it's psi sub S equals negative I CRT. This looks a little bit confusing, but let's try to keep it simple. I is the ionization constant. And you're going to need to know how to figure that out. And here's the key thing. If your solute is a sugar, anything that ends in OSE, sucrose, glucose, fructose, any kind of sugar, that ionization constant is always going to be 1. And that's because sugars don't break into ions when they dissolve. They don't ionize. So if your solute is a sugar, I is 1. That's important. Now, on the other hand, if your solute is a salt, the I is probably not going to be 1. Let's take the most simple case, sodium chloride. So when sodium chloride is put into water, it breaks up into ions. And I hope you guys know that it breaks up into a sodium ion and a chloride ion. So 1 formula unit of sodium chloride breaks up into 2 units of ions. So what would I be for sodium chloride? Somebody help me out. Since unit of sodium chloride to 2 ions. Let's look at a little more complicated one. So let's take calcium chloride. That's the formula for calcium chloride, CaCl2. When this dissolves, it breaks up into a calcium ion, which is Ca plus 2. And this 2 means 2 chloride ions. So one formula unit of this breaks up into 1, 2, 3 ions. So I for calcium chloride would be 3. Moral of that story, for sugars, your I is always 1. For salts, you have to break it up into ions and count how many ions form from the ionization of one formula unit of your salt. All right, C is the concentration. So let's go back to our formula here. C is the concentration. It's measured in molarity. All right, I see a question there from Talidra. Talidra, can you go ahead and ask your question?
Okay, so the solute potential of solution, the little box right here to give us the information on the side, will it be given to us on the test, or we have to remember this?
Yeah, so that whole little box is straight off the AP Biology formula sheet.
Right,
so you'll get that at the day of the exam, and you'll be able to use it throughout the whole exam.
All right.
Yes, ma'am. All right, so C is the concentration. That's the molarity of the sugar or the solute. That's going to be given to you in the problem. And we're going to work some sample problems here in a minute. But just know that C's concentration. It's the molarity. I'm going to skip R. I'll come back to R last. T is the temperature. This is where people get tricked up in these problems. The temperature has to be the Kelvin temperature. What's called the absolute temperature. And it turns out most of the time in the problems, they don't give you the Kelvin temperature. They give you the Celsius temperature. How do you go from Celsius to Kelvin? Who can tell me? How do you convert from Celsius over to Kelvin? Who can help me out? How do you convert from Celsius to Kelvin?
Add 273 to Celsius.
Yep. So Kelvin is equal to the Celsius plus 273. So just realize your temperature has to be in Kelvin, and all you have to do is add 273 to Celsius to get there. Now let's go back to R. So R is called the pressure constant. And that is given to you on the exam. It's this number. It's always this number, 0.0831 with some really strange units. Liter bars over moles times K. Again, this is going to be on your formula sheet. You don't have to remember it. You just have to know where it's at. It'll be on your formula sheet. That being the case, I want to go and show you a problem and how this works. So I'm going to work this first one for you, and then we're going to go back to Pear Deck and have you guys to try one or two. Before we do that, though, a couple other quick points. So the more solutes that are present in a container, the lower the water potential. And that's because as this C goes up, this whole number becomes a bigger number, but it becomes a bigger negative number, which means it's less, right? So like negative 14 is smaller than negative 13, right? So as this number part goes up, it becomes a smaller number because of the negative sign. And that's confusing to people. Remember that bigger negative numbers are actually smaller than smaller negative numbers. That confuses people. Some other couple things. To calculate the ionization constant, as I mentioned a second ago, to get I, for sugars, it's always 1 because sugars don't ionize. For salts, you break your salt up into ions and you count how many ions you get for each formula unit of salt that you started with. C again is the molar concentration, the molarity. R is that pressure constant. It's always that number, which you see right here on your formula sheet. And T is the temperature, It has to be in Kelvin, the absolute temperature scale. That being the case, here's a sample problem. So the molar concentration of a sodium chloride solution in an open container is 0.3 molar. This stands for molarity, right? The pressure potential of the solution is 0. What is the total water potential at a temperature of 25 degrees Celsius? All right, so before we can get the total water potential, we first need to calculate the solute potential. This is the formula we need to use. So to get the I, we look at our solute, sodium chloride. It's not a sugar, it's a salt. We know that because it's a metal and a non-metal put together. That gives us a salt. So we need to break that thing up, ionize it. And one of these breaks up into 2 ions, which means I is 2. So we're going to go -2. C remembers our concentration, our molarity, given to us in the problem right here. And remember that big M stands for moles per liter. That's what molarity means, moles per liter. All right, R is our gas constant, which is 0.0831. And remember, that's got weird units. It's liters times bars over moles times Kelvin. And our pressure, our temperature rather, tells us is 25 Celsius. We need that in Kelvin. So 25 plus 273 gives us 298. Now we look at our units, and we see right here that liters cancel with liters. Moles cancel with moles. Kelvin cancels with Kelvin. And the only unit that's left over is bars. So now we do the multiplication. So let's go -2 times 0.3 times 0.0831 times 298. And when we do that, we end up with a solute potential of -14.9 bars. Now remember that the total water potential is equal to the solute potential, which we calculated right there, plus the pressure potential. Well, they told us in the problem that the pressure potential is 0. So our total water potential is 0 plus -14.9 bars. So that means our total water potential is -14.9 bars. So we have completed the top question. The bottom question says, if a plant cell with a total water potential of -5 bars is placed into a beaker of the solution described above, will water move into or out of the cell? So what we're saying is that this solution in the container has a water potential of -14.9. But the solution inside the cell has a water potential of -5. And we want to know, is water going to move into or out of the cell? And remember that water moves from higher to lower water potential. Some of you are going to jump and say, well, it's 14.9 on the outside, 5 on the inside, water moves in, higher to lower. But you've got to remember that -5 is actually bigger than -14.9. Smaller negative numbers are larger than bigger negative numbers. So the higher water potential is -5. The lower water potential is -14.9. So the answer to that bottom question is that water is going to move out of the cell from where the water potential is higher, -5 inside the cell, to where it's lower, -14.9 outside the cell. All right, I want you guys to try one of these. So I'm going to get out of the PowerPoint. Going to go back to the Pear Deck screen. So go ahead and go back to Pear Deck with me. Let me move you guys ahead to the correct slide. All right, I want you guys to write on your screen and attempt to answer this question. So you've got the question at the top and the question at the bottom. So attempt to answer both by drawing on anywhere on your screen. It'll take about, say, 3 minutes to try to answer both the top question and the bottom question. Show your work. Try to answer both parts. All right. So I'm seeing a few answers starting to come in. I'm going to bounce back to the PowerPoint screen. Here's our problem. Whoops. So in this case, our solute is sucrose. Well, its name ends in OSE. Hopefully you know that means it's a sugar. Because our solute is a sugar, that means I is 1. Whenever you have a sugar as your solute, I is always going to be 1. So back to our solute potential formula, negative I CRT. In this case, I is 1. The concentration is the molarity, which is given to us, 0.4. And remember, that means moles per liter. Our R is always 0.0831. And again, that's liters times bars over moles times K. And then our temperature is given to us as 27 degrees Celsius. We've got to add 273 to that to get to Kelvin. So 273 plus 27 is 300 degrees Kelvin. Cancel out units. K cancels with K, moles with moles, liters with liters. We're left with bars. So we do the math. So -1 times 0.4 times 0.0831 times 300. So we end up with a solute potential of -9.97. It tells us in the problem that the pressure potential is 0. So our total water potential is equal to 0 pressure plus -9.97 bars of solute potential to give us a total water potential of -9.97 bars. Now, the bottom question says, if a plant cell with a total internal water potential of -7, so inside there, -7, is placed into this solution where the water potential is -9.97, what will happen to the cell's total internal water potential as time passes? So here's the thing. Water is going to move out of this cell, right? Because it's going to go from higher. -7 is a bigger number than -9.97. So water is going to move out of cell. But that's not what this is asking us. As water moves out, that means the solute concentration is going to get higher, which means this negative number is going to go down. So as as the solutes become more concentrated, the solute potential becomes a bigger and bigger negative number, which actually means it's getting smaller. So what it's wanting us to say here, as time passes, because water is moving out of that cell, the water potential is going to decrease. And that's because the solute potential is decreasing. All right, we got one more problem I want to look at, and we're about to run out of time here for today. So let's go ahead and skip this one, and let's look at this one. And we'll just talk through this one together. So sides A and B are separated by a semi-permeable membrane. So we're saying that there's like a cell membrane right there. Semi-permeable means some things can get through, some things can't. Assume that the cell's membrane is impermeable to the solute. So the solute in this case are these little triangles. So we're assuming these triangles are dissolved in water. If the volumes of the two sides are initially equal, so let's say we're in like a bottle or something. So the water levels here and here to begin with, they're pretty much equal. If the volumes of the two sides are initially equal and the membrane is impermeable to the solute, in which direction will water flow? Well, remember, water flows from where there's a higher water potential to where there's a lower water potential. So over here on side B, remember the total water potential is equal to the solute potential plus the pressure potential. So we've got negative 0.6 plus positive 0.2, or positive 2 rather, which means over here we have a total water potential of positive 1.4. On the other hand, on the other side, we have negative 1.7 plus 0. So we have a water potential over here of negative 1.7. So the water potential on side B is much greater than it is on side A. So water is going to move from B to A. That's the answer to number 1. Water moves from B to A. What is the total water potential on each side? Well, we calculated that already. Side B, it's 1.4. Side A, negative 1.7. Which side is hypertonic? Someone unmute and answer question 3 for me. Which side's hypertonic? And also tell me why you think that.
Side B.
Why do you think side B?
Wait. Because it has a lower solute concentration in side A.
Okay, but remember, what does hyper mean?
Hyper is high. So side
Hyper
A.
means high, right? So that means side A is actually hypertonic because it has a higher concentration of the solutes, the dissolved stuff. So side A, hypertonic, because it has more solute concentration. On the other hand, number 4, the side that's hypotonic, which means low or lower than, is side B, because it has less dissolved solutes.
Okay.
That makes sense?
Yes, sir.
Good. So question 5, what will happen to the volume of side A? Well, we just got through saying that water is going to move from side B towards side A. So if water leaves B and goes to A, that means the volume over here is going to go up while the volume over here goes down. So water is leaving B and going to A. So the volume of A goes up, the volume of B goes down. I saw a question there from Talidra again.
Oh, never mind. I thought you wanted us to answer it, but you answered it.
Oh, okay. Good, good. All right, let's see. The last question here is, why is the initial solute potential on side A lower than the initial solute potential on side B? So if we're just looking at the solute potentials. So it's asking, why is it lower on A than B? Well, remember the formula for solute potential is negative I CRT. Well, we're assuming that it's the same solute on both sides, which means I is the same. R is the same. It's always the same. And we're assuming the temperature on each side is the same, which means the difference in solute potentials has got to have something to do with C, the concentration. And what can we say about the concentration on side A compared to concentration on side B?
Side A is higher than side B.
Yep, side A is more concentrated. So as this number goes up, what happens to the solute concentration? The solute potential, rather.
Goes down.
C goes, what happens to the solute potential number?
It goes down.
It goes down because of that negative number in front, right? So as C goes up, solute potential goes down because of that negative in front of that formula. That's the key thing that confuses people. Concentration goes up, solute potential goes down because as that number gets bigger because of that negative, that means it's a smaller and smaller number. All right, I want to wrap this up by talking about one other quick thing, and this is water potential's relationship to transpiration. Who can tell me what transpiration is?
You said transformation?
Anybody? Just look at the picture. What do you think transpiration is? Yeah, it's the movement of water from the ground into the plant, up through the plant, and eventually out of the plant to back into the air. So it's how water moves through a plant. And it turns out this is all controlled by water potential. Turns out the water potential in the soil is somewhere around negative 0.3. But in the xylem, anybody know what the xylem is? Anybody able to tell me what xylem cells are? Those are essentially the cells in a plant that form like a pipe that transports water in the cell. So it's like the water tubes of the plant cell or the plant organism. So inside that xylem, the water potential is negative 0.6, which means it's high. The water potential is higher in the soil than it is in the roots. So water moves from the soil into the roots. Higher water potential, negative 0.3, to lower water potential, negative 0.6. In the trunk, the water potential is actually a little bit lower than it is in the roots. So it's negative 0.8 instead of negative 0.6, which means water moves from where the potential is higher in the root to where it's lower in the trunk. Water potential in the leaf, the cell, at least inside the cell walls in the leaf, is even a little bit lower. It's around negative 0.1. So that causes water to move from the trunk into the leaf cells. The air spaces around the leaf cells, still in the leaf, but in the air spaces around the cells, that water potential is even lower, negative 7 compared to negative 1. That means that water moves from the cells into those air spaces. And then the water potential outside, in the air outside the plant, the water potential out there is like negative 100. So it's way, way lower. So water moves from those air spaces in the leaves out into the air. Point being, water is always moving from higher water potential down here in the soil to lower water potential out in the air. So the whole movement of water through a plant is controlled by water potential. That's where we're going for. That's something that I could see being a good like free response question or something on an exam. All right, I've thrown a lot at you today, so I think I'm going to stop there. Questions I can answer for you at this point? There is a whole video in our A+ resources that goes through these water potential calculations. So if what I did today in a hurry confused you, ask your teacher for the link to the water potential practice problems, and it'll walk you through step by step how to do all these calculations with lots of explanations. All right. Otherwise, I hope this helped. Anybody have a question? Go ahead and ask. All right. Well, thank you guys for being here today, and we'll do this again. We won't have this next week because we're having our normal A+ study sessions. So the next one of these will be week after next. Still Monday at 4 o'clock, but it will be 2 weeks from today. Thank you guys
All right,
again.
thank you.
Thank you.
Thank you.
So we should expect the responses from this pear deck to be in our Google Drive or in our email.
It should be in your Google Drive account under shared with me. And that should happen within the
All right, thank
next 20 to 30 minutes.
And if it doesn't work, just contact our teacher.
Yeah, just ask your teacher to email me and I will send it directly to the teacher.
Yes, sir.
All right. Thank you guys again.
Thank you. Take care.
You too.